beautiful people. Welcome back to Best Medicine's GCSE AQA Biology lecture series. Today, we're going to be talking about variation and evolution. How does variation give rise to evolution? And essentially, evolution is the ability of, or the concept of organisms becoming more and more complex and more adapted to their surrounding over time, developing newer features so that they are more suitable towards serving their role in the environment. So, we have been discussing variation for a very long time now. We started with mitosis and meiosis, and we said mitosis produces identical cells, while meiosis produces genetically different cells. And then fertilization further plays a role in enhancing genetic variation. Mutations also give rise to different genes and different alleles, giving rise to variation. So variation essentially is just having different genotypes which give rise to different phenotypes. So variation stands for organisms having different differences in their phenotypes due to variable genotypes. For example, you could have rabbits, and rabbits having different colored furs, right? The gene for the fur has different versions or alleles, giving rise to variable colors of fur. So where does variation come from? Variation can occur due to either the influence of environment or of genes. So environment influence can kind of work in this way, is that you have I'll give you an example. Let's say you have twins, right? You have two twins, and one of them goes to the gym and the other one does not, right? The one that goes to the gym has also a good diet, and he has a good mental health practices that he does. He takes care of his mental health, his physical health, and so on. While the other one is just laying around all day on their TV and doing nothing, and just not working on themselves. And so you'll have major differences in both the physical as well as mental qualities of the two twins. So that is a good example of environmental influences, right? Environment can definitely affect how the organism eventually develops up to be. And then, of course, we have genetic variation. Genes, different alleles, different versions of alleles, of course, give rise to different proteins. And then those proteins will code for different or perform different functions. And then those different functions give rise to variable qualities. So, genetic variation. I want to read this through. Let's say there's only white fur rabbits in an area, right? How do these white fur rabbits eventually develop different colors? That's a good question, right? The answer to this is mutations. Now, we've already done this, and because you've been following this particular series for a very long time now, you have a good idea of everything that's going to be discussed today, hopefully. So, mutations eventually cause a change in DNA bases, possibly resulting in a change in phenotypes. And when we have a change in phenotypes, that gives rise to different alleles. For example, I'll give you an example, right? We have a box, right? We have an area, and in this area, we only have white fur rabbits. It's that example again. So if we only have white fur rabbits, and all of a sudden we get this mutation in which one of them sort of produces an offspring that has brown fur, right? So this is due to a mutation. Mutation in the color gene, fur color gene, could have occurred to develop a completely new offspring who has a different fur color. Now here's the thing, this mutation that occurs, for it to become part of the species, it has to be inheritable, and that's very important. The mutation has to be inheritable, which means that the mutation at least has to go into the germ cell, right? Remember, the germ cells, usually, at least in sexually reproducing organisms, it is the germ cells that eventually give rise to offsprings. And so the mutation has to be inside the germ cell to be able to be inherited. I'll give you an example of mutations that are not inheritable. For example, somebody gets cancer, right? Cancer is a mutation. So many people get cancer. It doesn't mean that when they have children, their children are going to have cancer at birth. That is because the cancer mutations are not inheritable, because those are not in germ cells. Those are in somatic cells, right? So somatic cell mutations do not lead to evolution, do not cause genetic variation all that much, as compared to germ cell mutations, which eventually play their role in evolution. Okay? You can also call this meowtation. When a DNA gene is damaged or changed, meowtation occurs. Okay, Next up, we have environmental variation. So, of course, genetic variation includes mutations, but what about environmental mutations? So again, the example of twins would show up. So let's say we take two identical twins at birth and send one to a hot region near the equator in an underprivileged household and the other one to a cold region with a well-off household. So if these are white twins, then one would have a darker skin, the one that is present in a hotter environment. Why? Because he's constantly exposed to the scorching sunlight, and so his body will adapt and it's going to start producing more melanin, more pigment on the skin to protect itself from the UV rays of the sun. So also because he is going to be living in an underprivileged household, he's going to have a poorer physical health. While the other one has a lighter skin and better physical health because he's present in a colder and more well-off household. So environmental variation, obesity, sedentary lifestyle versus exercise and healthy diet. We've already looked at these examples in cardiovascular disease as well as in diabetes. So these can increase the risk of development of diabetes, coronary heart disease, and therefore this is also a kind of environmental variation. And so this variation eventually gives rise to different kinds of versions of species of the same species in a given area. And some of those species that are more adapted to the surrounding are going to be better suited to survive a selection pressure. So if a selection pressure comes in, for example, something tries to kill, let's say a disease comes in or a predator comes in and it kills off these people here, well, the yellow one still survives, maybe because it has a better camouflage. Whatever the case is, when this survives, that is called natural selection. Nature is selecting this particular individual to proliferate and eventually become the new species. So that is called evolution, and we're going to dive into it now. So theory of evolution. Before we go deep into evolution, we have to understand the pioneer. Evolution, the theory of evolution, was presented by Mr. Charles Darwin. Charles Darwin was a scientist who essentially gave the theory of natural selection, evolution by natural selection. And what he did was he had a big research. He went to the Galapagos Islands and he had voyages throughout the world, and he looked at the differences and similarities between different species, things that he could observe with his naked eye, and gave up his theories, sets of theories, and he wrote a paper and wrote a book about essentially evolution and natural selection. So what he did essentially was that Darwin traveled to the Galapagos Islands and he observed the differences in different animals between the islands. So let me just quickly explain this to you. You have the sea, right? And then the sea has a couple of islands nearby. So here's one island, another island, and so on. So we have a couple of islands nearby, and then each of these islands are going to have their own different environments, right? This one might have different fruits, this one might have different foods, right? Different food, and so on. So for example, if you have a couple of birds that migrate here, let's say finches, right? So we have a finch that is a parent finch, and then this finch, a couple of them go there, they're migrating, and a couple of them go there, and a couple of them go there, and some go there. So what would occur is that each of these finches, as they go ahead and start proliferating in their given environments, the environment is going to select the best out of all of them. So basically, for example, if you have softer fruit here, right? Softer fruit as compared to, I'm going to do this one and I'm going to do this one. So softer fruit over there, and then we have thick shell fruit or thick shell nuts here. So if you have thick shell nuts island and you have a softer fruit island, but the softer fruit island is going to select the finches that have a delicate beak, right? Delicate beaks. Why? Because the fruit is very easy to be pierced. But for the island that contains thick shell husks and nuts and stuff, then for that you will need finches that have a thicker and a stronger beak. So stronger beak finches will be selected for this one. Okay? Similarly, we could have tortoises, right? Or turtles. And the turtles that live in an area that has grass, okay, let's select these two islands. So this one maybe has high grass, right? And this one has low plantation. So the one that has high grass, the necks of the turtles are going to be longer necks. And next of the turtles over here are going to be shorter because there is low plantation, and the turtles would not have to be adapted to reach the higher plantation. So that is so what happens eventually is that these species start becoming so different from their parents that eventually a point comes in which they're not able to breed. Okay, so maybe let's say this is time zero, right? Time zero. I'm just gonna give you a timeline. At time zero, they migrate. So maybe 100 years later, they have a little bit of differences in them, right? Maybe 1,000 years later. So maybe 1,000 years later, you have finches that have become very different from each other that they're not able to interbreed even, right? So 1,000 years later, they're so different. We have a finch on one island, we have a finch on the other island, and they're very different that they're not able to breed with each other. Maybe after 100 years, they are able to breed with one another, but after 1,000 years, they're not able to breed with one another. And so we would say that, okay, they have become completely different species. So the finch, the parent finch, at time zero has now, after 1,000 years, given rise to two different distinct species, and we would say that it has become evolved. Okay, so anyways, let's have a look at some examples. So, number one, we have finches. So finches on islands with tougher shells and seeds had bigger and stronger beaks. That's what he observed. While finches on islands with fruits and softer food had thin beaks. For example, number one, very thick beak, right? Number four, thin and delicate beak. Number two, turtles with higher grass had longer necks, right? Turtles with higher grass had longer necks, and turtles with lower grass had shorter necks. Lizards were similar colors as their environments, and so on, right? So that they're able to better camouflage themselves. Okay. Here's a meme. Hey, evolution. Yes, what do you want? And then he's disappeared. Do you know who this is? That's a dodo bird. A dodo bird is a very popular extinct bird. So, together with knowledge of how earlier fossils were less complex while newer fossils were more complex features, and other knowledge available at the time, Mr. Darwin, using also his own observations, gave this following theory and made our life hell, which is that there is variation between organisms of the same species. And this variation means that some are more adapted, and so therefore they will be selected towards a given environmental selection pressure, while others are less adapted. The more adapted ones have a greater chance of survival to reach reproductive age. So once they have a greater chance of reaching reproductive age, they have a greater chance of reproducing, and therefore a greater chance of producing offsprings. So as they reproduce, the well-adapted trait gets passed on to the offspring, and the less adapted one does not. Okay, so while the opposite happens to the less adapted. So number of more adapted organisms increases while less adapted decreases. And eventually all less adapted trait containing organisms die off, leaving behind only well adapted ones. And at that point, when we have complete dissolution, complete removal of the original species, and only the new trait, the new ones, the evolved ones are there, we say the species has evolved. Okay? And again, that variation has to be obviously be inheritable. So let's quickly try to describe this using an area here. So again, let's take the rabbits as an example, which is a good example, I guess. So you have rabbits, and these rabbits live in a brown-colored environment with brown leaves and brown grass, and it's very easy to spot them by the predators because these are white. Right, a predator comes in, it's going to be very easy to spot these. But what happens is that one of them gets a mutation. Okay, maybe this one gets a mutation, and once this one gets a mutation, he produces a brown fur-colored rabbit, which is able to better camouflage itself in this particular environment. So now what occurs is that the red predator is going to come in and start killing them, right? Start killing the original species. So these original species, even though they are reproducing and maybe making little offsprings, those little offsprings do not have the chance to reach reproductive age because they're quickly being killed off by predators. Okay, while the more adapted ones have the chance of reproducing and have the chance of reaching reproductive age. And so they start increasing, while the other ones start decreasing because they have lesser chance of reaching the reproductive age. And eventually, all of them, all of the original species are wiped out, and only the new ones remain. And that is when we say the species has evolved. Okay? Now, what is speciation? Speciation is when a new species is formed, right? An example of this could be this one here. You'd have a new species if they're not able to interbreed with one another. I'll give you some examples, right? I'll give you some examples here. You know, a Labrador, right? And a Rottweiler. If you cross them with each other, it will produce a dog. And that dog can go ahead and give you more dogs. Okay, so that dog is fertile. It is fertile. This means that these two are the same species. Same species because they're able to interbreed with one another and they're able to produce a fertile offspring, right? These two conditions have to be met for something to classify as being part of the same species. Number one, you have to have the ability to reproduce with that with the species in between the species. And number two, you have to be able to produce a fertile offspring. So those two conditions have to be met. Now let's have a look at a lion, right? And another lion. Of course, a lion and a lion is going to produce a lion, and that lion is going to go ahead and produce more lions, right? And a tiger is going to produce, it's going to mate with another tiger, and then it's going to produce tigers, and that tiger is going to go ahead and give you more tigers. Very simple and straightforward. But here's something that can also occur, which is that you could have a lion plus a tiger. Now, when they reproduce, they produce something called a liger. That's the largest cat in the world. But ligers are not able to mate with other ligers and produce another liger. Okay, they're not able to reproduce. They're infertile. Because these are infertile, one of the conditions is not met. So because of that, lions and tigers are different species, separate species. While Labradors and Rottweilers are the same species. Number one, because they are able to produce an offspring, and number two, that offspring is also fertile. So that is speciation. Speciation is when once. Let's make another example. Okay, we have an organism, and that goes and migrates to two areas, right? So here's a mountain, and then you have two areas. Area 1, you have Area 2. Now, Area 1 is going to have its own selection pressures, right? Area 2 is going to have its own selection pressures. So that organism, when it gets adapted and it gets naturally selected, over time becomes more adapted to this area. And this one, the same organism gets adapted to this area over time. Eventually, so much differences occur between them, right? Initially, let's say the organism is kind of like a triangle. Here, right? Initially, it's like a triangle, and then that triangle went over there, and then that triangle went over there, right? And it's kind of becoming now this, and this one over here is kind of becoming like a star, and then eventually this starts becoming pink, right? And then this one becomes a star. So maybe at this point they're able to interbreed with one another and produce a fertile offspring. Over here as well, maybe they're able to interbreed with one another and produce a fertile offspring. But at this point, they're not able to do so, and therefore we say that they have become completely different species because of different selection pressures over time, right? So one species has given rise to different species over time. Why? Due to natural selection through evolution through natural selection. So that is speciation. Let's have a look at this very quickly in neat notes. So speciation is the development of a completely new species. Okay. This occurs when organisms evolve and become so different from their original ancestors that they either, number one, cannot interbreed with them, or number two, they can interbreed, but then they produce an infertile offspring. Okay, cannot interbreed, for example, a bee and a crocodile are obviously completely two different species because they cannot interbreed. But if they can interbreed, then they produce an infertile offspring. For example, a horse and a donkey make a mule, and a lion and a tiger make a liger. And both mules and lions, ligers, are infertile. So here's a meme on speciation. I'm sorry, my friend, even though you won't probably won't believe me. It's not you, it's me. When they look similar but a little bit different because they have become different species, so they're not able to produce children. What do you mean I'm not a bear? I have all the qualifications. Okay, and next up we have extinction. Extinction is quite straightforward. I think you guys are already aware of what this is. It is essentially the permanent disappearance of a species from Earth, like the dodo bird, right? The dodo bird is extinct. This occurs when the last individual of that species dies. So essentially it signifies the end of species' ability to sustain its population because of very severe selection pressures. Some famous extinct species include dinosaurs, which were killed by obviously meteors that we theorize, dodo birds, mammoths, Tasmanian tigers, and so on. Now, what are some of the causes of extinction? Now, that's high yield, and you're supposed to know this. And I think you can pretty much intuitively also be able to think this out. Habitat destruction, if you don't have a place to live, obviously it's not going to be good for the species and can go extinct, right? We'll talk about deforestation as well. Climate change, again, you kill the habitat and get rid of the food. Overexploitation, for example, overfishing. Invasive species, you could have, in Australia, you had the problem of rabbits, which were invasive. So invasive species can also kill off, due to competition, kill off the resident species. Pollution, disease, and natural disasters. For example, meteor killed all the dinosaurs, right? Another famous question that shows up on the exam, I put this here just so you're aware of this, and hopefully it will show up on the exam so you can get on the exam again so that you can get full marks. Why did people not agree with Darwin? Why did they have disagreements? Well, number one, there were different religious beliefs. Okay, people used to believe, for example, that humans come from Adam and Eve. That's a popular belief, right? I'm not saying it's wrong. It could be right, because Darwin's theory is still also a theory, right? It's not 100% accepted fact. Lack of supporting evidence. For example, Darwin could not explain where variation in traits comes from. There were other more attractive theories around that people liked more. For example, Lamarck's theory. Especially there was lack of supporting evidence. So if a question on this comes for 2 marks, then maybe I will go for this one and this one. Okay? So who was Lamarck? What is this? What was Lamarck's theory? Well, we have to know this for AQA, so let's have a look. Lamarck, or Jean-Baptiste Lamarck, was a scientist who proposed that animals can pass on traits that they acquire during their lives to their babies. That's a problematic thing. It's kind of like having cancer and then you're able to pass it on, right? That's what he said. He said that if you acquire a trait in your body, in your physical body, you're able to pass it. And as you pass it on the baby, and the baby eventually also works on that trait and improves it, and then they pass it to the next baby, and the next baby inherits the improved version, and then the next baby sort of improves it in their life, and eventually pass it on to the next baby, and so on. So kind of like having giraffes, right? So you have a giraffe and it's trying to reach for this tree, and then it works a little bit, right? Maybe it extends its neck a little bit, and then it has an offspring that has a longer neck, and then the other offspring has even a longer neck, and the other offspring has even a longer neck. Now that's problematic and it's completely wrong, okay? You have to understand that this is a wrong theory. This is not correct. Why? Because these traits that you acquire during your life are not inheritable, okay? Unless the mutation is in the germ cell, that is not inheritable. It's kind of like you go to the gym and get big muscles, right? If you get big muscles, it's not like your child is going to automatically have bigger muscles, right? It makes no sense. So Lamarck's theory was wrong. and this was rejected. Um, Only traits that people get from their genes and that are inherited from their parents can be passed on to their babies. So Lamarck's theory, you have to understand what this is. You have to remember the name Lamarck, and you have to understand that the theory is wrong, but it is one of the reasons why Darwin's theory could have been rejected at that time. And next up, you have selective breeding. Do you remember natural selection, right? We talked about natural selection being the ability of nature to impose selection pressures and select the best adapted species in a given environment. Well, we can do the same thing, just artificially, right? Just grab the best species by ourselves and have them mate. I could grab the best milk producing cows and the bull that produces the most milk producing cows and get them to mate with each other and eventually get myself a lot of high milk producing cows, right? That's a good example. So selective breeding is the process of choosing specific plants or animals to breed together to produce offspring with desired traits, like bigger flowers or more better fruit, right? So this is kind of like artificial selection because you're doing it by force artificially. Examples of this would be breeding dogs for specific traits, like, for example, Labradors are for friendliness. For developing high yield crops, like wheat. Breeding cows for increased milk production. So, what are some of the advantages of selective breeding? Well, of course, it enhances the desirable traits, increases productivity and efficiency, and also it can produce disease-resistant plants and animals, and we'll have a slight look at this in a bit. Disadvantages of selective breeding. It reduces genetic diversity. Why? Because you're constantly breeding the same thing with itself, right? So you're not having a lot of genetic variation, which means that you're kind of like having clones of the best species, best variation of that species. So a selection pressure is more likely to be able to wipe out the similar species. It can also lead to inherited health problems, and it may result in unintended negative traits. It's kind of like having consanguineous marriages, right? Cousin marriages. You're constantly breeding with one another, and it increases the likelihood of developing autosomal recessive diseases. Remember autosomal recessive diseases? If you have a autosomal recessive disease running in the family, and you keep on marrying inside your family, there's a good chance that eventually one of the offsprings is going to get that disease. But if you keep on if you start marrying outside of your family, which does not have that disease running in their genes, then there's a lesser likelihood that your offsprings are going to have the disease. Similar case here, that it can result in unintended negative traits because you have you're breeding the same species and cousins essentially with each other. Here, maybe I'll get some food at the campfire. What's the worst that could happen? 10,000 years later. Selective breeding to select the best wolves, the most friendly wolves. And eventually we get to this. Artificial selection. It is the selective breeding of plants and animals to promote desirable traits of offsprings, okay? Next up we have genetic engineering. Now, because we already talked about this in the previous video, I think, it is essentially obtaining desirable genes, like desirable enzymes or desirable proteins, and genes for those proteins, and then putting them in another genome. So just a review of this, basically genetic engineering transfers genes between organisms. You can have a beneficial gene from one organism to another to achieve a specific trait. What you do essentially is that you extract the useful gene first. How do you extract it? And you need to know this particular enzyme name. You use a restriction enzyme, kind of like a scissor that's going to cleave the useful gene, and place it into a vector, either a virus or a bacterial plasmid. And then that vector delivers the gene into the target organism cell. Let me just quickly explain this to you here. Do you remember cystic fibrosis? Right? Cystic fibrosis was a disease, autosomal recessive, in which goblet cells are producing a lot of very thick mucus, right? So these are producing very thick mucus here, and this is a goblet cell, and it contains this DNA here inside the nucleus. Now imagine we take the correct gene, right? We get the correct gene from somewhere. Let's say here's a person's DNA, right? And then in this DNA, here's the correct gene. What we do is we take this gene out using restriction enzymes, kind of like scissors. So we take the gene out, we'll have this gene, and we put it inside a virus, let's say, right? Maybe HIV or something, a virus that takes it to the respiratory tract. So next thing we do is we take a virus, here's Mr. Virus, right? And we placed the correct gene for which will cure cystic fibrosis, which is mutated in cystic fibrosis in this virus. And we send this virus to go do its thing, right? Go infect the cells, do your thing, and put the gene in there. So remember how viruses work is that they install the genes into our machinery, into our cells. For example, HIV. HIV would install its genome into our DNA. So here's what this virus is going to do. This virus is going to go ahead and insert the correct DNA into the nucleus over here. And when it becomes part of the nucleus, now this cell starts making the correct protein. You understand? So that is an example of gene therapy. And that this virus here that is carrying the gene to the genome over there to the cell is called a vector. Just like we had vectors for malaria, right? Vectors in diseases. This is a vector that transmits DNA. And that is used in genetic engineering. The other example that we did in the previous video, in one of the videos that we did, was transferring or making insulin, right, using bacteria. So a couple of diagrams here, the restriction enzymes, they remove the bases, remove the genes, and then you're able to plug in those genes into the desired cells. A very important example that you need to be familiar with is the example of golden rice. Now here's the thing, back in the day in China, where people they eat a lot of rice, right? They eat a lot of rice, and all they plant is rice in a particular area. So because they were planting rice all the time and eating it all the time, they started having these vision problems, and scientists discovered that they had severe, on a mass effect, they had a severe vitamin A deficiency. And so what they did was they engineered the rice to become golden rice, and then the government instructed them to, instead of planting white rice, just start planting golden rice. And the golden rice, the gene for vitamin A was put in the golden rice, and the people started eating golden rice, and the deficiency went away. So that's an important example of genetic engineering that often shows up on the exams. They would not directly ask you what this is, but they could put that in a question, and the more you're familiar with this, the more you'll be confident about the question, okay? The golden rice is a rice engineered to produce beta-carotene, a precursor of vitamin A, and you need to know this word, okay? Beta-carotene is vitamin A precursor to help combat vitamin A deficiency. So examples of genetic engineering include bacteria engineered to produce human insulin for diabetes treatment, and we talked about this earlier. Genetically modified crops that are resistant to diseases, right? And we can also have sheep altered to produce medicinal substances in their milk. Or we can have gene therapy, for example, treating cystic fibrosis in this way. Now here's the thing, genetic engineering is not a straightforward topic. It's a controversial topic because, again, you're playing with nature. Because it's a relatively new field, there are a lot of concerns about its long-term impacts and unforeseen issues because we don't know its long-term effects because it's a very relatively new field. There's also opposition from people because it may be seen as playing with nature and its normal way of evolutionary development. So, next up we have GM crops, right? We just talked about genetically modified crops that are, for example, resistant to diseases, insects, and herbicides. I've made a separate section on this because this occasionally comes up on the exam, on the AQA exam. They can ask you about the pros, but most importantly, they will ask you about the cons of GM crops, and that's quite important. The cons are very important. So let's have a look. Some of the advantages include disease resistance, right? For example, you could have tobacco mosaic virus resistant crops. You can also have combating nutrient deficiencies, and we just looked at a good example of this. For example, golden rice that is enriched with beta carotene, vitamin A precursor, and higher yield crops, of course, giving you better efficiency, better yield, better productivity. So these are some of the advantages. What are the disadvantages? And this is a popular question, almost shows up every exam. And the answer to this is, of course, because you'll be planting the same GM crop in a very big field, it can result in reduction in farmland biodiversity. Number two, safety concerns, because we have an incomplete understanding of GM crops' impacts on human health, for example, including potential allergies. There's also, and this is the option that I would go with first, and very popular, is that these GM crops, right, they can go ahead and breed with wild plants, transferring those genes to wild plants. So for example, if you have TMB resistance and you transfer it to a wild plant, now the wild plant also becomes TMB resistant. We don't want the wild plants to become TMB resistant, right? And spreading into natural environment, creating super weeds. And also, traits selected for GM crops may not always be ideal, right? It could possibly increase yields, but also make the food more calorific, increase the calories. However, let me remind you that genetically modified crops are already in use in many areas without major issues. But this is something that often shows up on the exam, so maybe you should write this one first. Okay. This one second, maybe this one third in your answer if it shows up. Okay, we have a meme. Nature creates viruses to save world from overpopulation. Humans cure diseases with viruses. Cure diseases with viruses. For example, cystic fibrosis, right? We just cured it with a virus that served as a vector. You let the virus do its thing, transfer the DNA into the goblet cell. Next up, we have something called cloning. We've already done cloning a couple of times, so I'm just gonna very quickly go over this. Remember, the plants have the ability to essentially undergo mitosis and produce undifferentiated stem cells. So any cell on a plant, almost all plant cells, like a leaf cell or a root cell, or even meristematic stem cells from meristems, like growing regions, have the ability to essentially give rise to undifferentiated stem cells. And then those cells can become an entirely new plant. Okay? So there's two ways really to clone the plants. One way is that we take plant cuttings, usually from growing regions like meristems. And we take that, we plant that plant cutting in the soil, give it proper nutrition, light, and so on, and becomes an entirely new clone of the original plant. The other way is to culture the meristematic stem cells. So we take stem cells and then we culture them, and then these eventually grow up to small plants, and then we can plant them into pots. Okay? It's very simple, very easy. However, in animals, it's quite different and difficult. Why? Because in animals, we don't have these specialized stem cells, not specialized, undifferentiated, unspecialized stem cells that are able to become any kind of cell, right? In plants, that is the case, but in animals, we don't have that case. In animals, we have stem cells that are already partially specialized, and they can only form a couple of types of cells. For example, in humans, we have bone marrow cells, right? And they can only form blood cells, and so on. Well, so how do we clone animals if their cells are already specialized? It's very simple. Here's what we do. Cloning in animals is done by acquiring sperm from the desired male and eggs from the desired female. And then we allow them to fertilize. What we get is a zygote, and we allow the zygote to divide a couple of times, okay? And we get a cluster of cells. So once we have a cluster of cells, we take the individual cells, the zygotic cluster, and then we're going to go ahead and plant them in different embryos, okay? What I mean by this is that here's the sperm from a bull, right? Here's a bull sperm, and then here's a bull egg. So this is a bull sperm, and that's a bull egg, and then they eventually give a zygote. And then that zygote is going to go ahead and develop into a cluster of cells, right? Many different cells. This cluster of cells, each of these is an embryonic stem cell. So we can take these individual cells, and these can function as a zygote, right? As a single zygote. Each of them can give rise to a new offspring. So we take these cluster of cells, individual cells, and plant them in cows, right? Cow 1, for example, right? Cow 2, cow 3, cow 4. So these cows are not the original mothers of these cells, but we plant them in these cows so that they serve kind of like surrogate mothers, right? Kind of like surrogate mothers, and eventually give rise to the clone of the best bull and the best cow, even though we have different cows that are giving rise to those offsprings. Okay, so that's one way of cloning. Another way of cloning is called adult cell cloning. Adult cell cloning is that we take a female egg and remove its nucleus, and then take a somatic cell and obtain its nucleus and inject it in the female egg cell. What did I just say? That makes no sense. Let me just explain this to you, okay? Remember that you have a female egg cell, right? Female egg cell has how many chromosomes? N or 2N? It is N number of chromosomes. The female egg cell has N number of chromosomes. What we want it to be is a 2N number of chromosomes so that it can become a zygote. Remember, a zygote has 2N number of chromosomes, isn't it? That's a zygote. So how do you get this to become this without a sperm? Here's what you can do. You can take a normal somatic cell, right? Here's a normal cell, maybe from some gland, right? Or the breast or something. We take a normal cell that has 2N number of chromosomes. Here's what we do. We cut this nucleus out, we take the DNA, right? And we remove this nucleus out. Replace the nuclei from a normal somatic cell into the female egg cell. So what would happen as a result is that this is gone now, and we are putting the 2N nucleus into the egg cell. When you put the 2N nucleus into the egg cell, it has now become a lot like a zygote, isn't it? So what we have done essentially is that we have completely taken exact same DNA from an organism, an animal, and we have made a zygote that has exactly the same DNA as that animal, a clone of that animal, right? A zygote clone of that animal. And we can allow this zygote clone to eventually start dividing by putting it into an embryo. So maybe we put it in an embryo, it becomes a cluster of cells, and eventually becomes an entire new organism. And this has been done before. The first clone that was made in this way was Dolly the sheep. Let me show you very quickly. Here's Dolly the sheep. So that was the first adult cell clone. Okay, so let me review this very quickly. Adult cell cloning is that you take a female egg cell and remove its nucleus, right? That is the future zygote. You remove the nucleus of the female egg cell. Then you take a somatic cell, like for example from the mammary glands, and obtain its nucleus, and then inject it in the female egg cell that has lost its nucleus. The female egg cell now becomes a zygote, right? Just like a fertilized zygote. And then we stimulate the zygote to divide and take the resulting embryo and implant it into some host. And eventually the host will give us an organism. So Dolly the sheep was the first adult cell clone prepared this way from a mammary gland cell. Okay? Of course, there's issues related to cloning, like there's issues to everything. There's obviously religious issues, religious and ethical concerns of playing with nature. The resultant clones occasionally have several health defects as well. And cloning reduces the gene pool by producing identical organisms, making a species more susceptible to selection pressures as well. And there's also another concern that because you're making this cluster of cells, you're essentially people have concerns about torture, right? So that's an ethical concern. And yeah, so that's it for this particular lesson. I'll hopefully see you guys in the next one. And if you have any confusions, anything that you don't understand, you can rewatch the videos. If you still don't understand, then leave a comment or send me a message and we can probably book a lesson with a tutor, okay? I'll see you guys in the next video. Take care, have a great, wonderful study session and review this really, really well.